Good morning and thank you for joining us today for the Ameria Limited Investor Webinar. My name is Jane Morgan and today we'll be discussing Emyria's novel MDMA-inspired drug discovery program and world-leading partnership with the University of Western Australia. Emyria's Managing Director, Dr. Michael Winlow, and UWA's Associate Professor, Dr. Matthew Piggott, will present an update into the developments of their joint MDMA-inspired drug discovery program aimed at developing MDMA-like drugs with the potential to treat psychological disorders, including the recently announced collaboration with Psychedelic. I want to hand you over to Dr. Michael Winlow.
Thank you for joining this morning. A real pleasure to shine a spotlight on our MDMA-inspired drug discovery program, an activity we've been working closely with the University of Western Australia on for the last 12 months, and it's come a long way, and a real pleasure to show you what we have accomplished and where we want to go next. So this morning, we'll go through a quick introduction to Ameria. We'll explain the growing interest behind psychedelic-assisted therapy, which is really drawing a lot of investment and interest into these compounds. Then I'll hand over to Dr. Piggott to talk about what is MDMA, take us through some of the therapeutic applications of MDMA itself, but then go in deeply on our MDMA-inspired drug discovery program, where we're using MDMA as inspiration to develop novel compounds that have the potential to treat a range of diseases. And then back to myself to give an update on the results so far and where we hope to go next before going into Q&A. So Ameria is a clinical stage biotech company. We're focused on unmet needs in neuroscience and mental health, but we're unique in that we spend a lot of time learning from the experience of real patients that come to see us in our clinical service subsidiary, Emerald Clinics. So we have two really progressive leading programs. Our clinical program, which is already into phase 3, is an ultra-pure cannabinoid delivery technology that's generated multiple dose forms, the first of which EMD-RX5, now in phase 3, I mentioned, and we're progressing a number of global registration opportunities with that dose form. We also have a substantial R&D preclinical pipeline from the MDMA-inspired medicine discovery work that we're doing with the University of Western Australia. And what makes us unique, I think, as a biotech is not only have we made tremendous progress, we're already in clinic on these initiatives, we have access to an enormous proprietary real-world database that we gather with our patients from our clinical service subsidiary, Emerald Clinics, and that's guided where our clinical programs have progressed. But we also have now this unique drug discovery program through our partnership with the University of Western Australia and an amazing global team of collaborators and in-house experts who have done drug registration before with multiple FDA successes. So that really touches on 5 key highlights, which set our company apart. We've got this unique cannabinoid delivery technology that's partnership ready, that has some really unique advantages over the incumbent approaches to delivering cannabinoid medicine. And those insights were inspired by the real world data that we've gathered with our patients and our firsthand experience working with these medicines for many years. That's allowed us to have a number of programs already in the clinic, RX5 being phase 3, and RX7 being our prescription cannabinoid medicine approaching phase 1 trials. And to that resource, those assets now, we've added a substantial R&D pipeline with one of the world's largest libraries of MDMA analogs. That's the focus of today's discussion. Of course, we've got a terrific team as well. So we think we're really well prepared to capitalize on these great innovations. And so our vision now is to accelerate the development of really promising treatments, but where the evidence has been lacking, by generating data with patients and working with global experts to develop new treatments, both formulations and novel chemical entities to tackle those unmet needs. We are continuing to advance our cannabinoid program, taking that into the clinic, into registration opportunities globally. But now we have this opportunity, this really exciting potential to become a global leader in the development of neurological treatments inspired by MDMA. We'll explain why that compound is such great inspiration for us. So a little bit about this growing interest in psychedelic-assisted therapy. It's getting a lot of attention now. There's a Netflix documentary. You may have done your own research and seen the media attention into this therapy space. And not surprisingly, it's motivated by this growing recognition of this global mental health burden that is becoming more apparent and heavier, really. And just to highlight sort of 2 major conditions where psychedelics are starting to show promise in being heavily researched, that's in post-traumatic stress disorder, which incredibly affects 2 out of 3 veterans and can have a remission rate as low as 5% with standard treatments. So clearly a major unmet need there and a massive burden. Major depression, another space where psychedelic assisted therapy is showing tremendous promise. Again, a condition where current treatments, do not help everybody, and also have a number of known and unwanted side effects, which limit sort of the utility and benefit of these treatments. So with this being the state of the world today, not surprisingly, people are looking at new approaches to tackling these serious conditions. And so psychedelic assisted therapy is starting to get a lot more attention. And so what it is at its core is the use of psychedelic medicine that can come from a long list of plant-inspired compounds and also some synthetic compounds. Names that we probably recognize, LSD, MDMA or ecstasy, fall into that category, psilocybin as well. And these compounds, when given to patients, can change the way the brain communicates internally and therefore break patterns of rigid thinking, ruminations, create different ways of feeling and behaving that can make psychotherapy more effective and hopefully more durable for those patients as well. But it's also believed that these compounds can also affect the brain directly, inducing plasticity or the growth of new neurons, and help establish new pathways as well, and ultimately lead to really durable long-term benefits for patients. And we're just at the early stages of researching these compounds. Given the state of the burden, the promise of these medicines, not surprising that there's tremendous investment and interest in companies that are innovating and pushing the frontier of this space. A number of major companies, most of these based in the US. You can see many of them have novel development programs across a range of compounds as well, as this list shows. And Emyria is no different. We're really pushing the edge here, but staying pretty focused on MDMA and its derivatives as our source of inspiration and new medicines. I'll hand over to Dr. Pickett to take us through what is MDMA and a little bit about how our program is proceeding.
Thanks very much, Michael. Great to be presenting today. First question here, what is MDMA? MDMA is synonymous with ecstasy, which a lot of people have probably heard of. It's a drug that's used recreationally, has been by millions of people around the world. It is important to recognise, though, that what is sold as ecstasy is not always MDMA. That's why I always tell my kids and my students I would never buy drugs on the street, because even though you think you're buying ecstasy, there's a chance you might be getting something else. And some of the substances that have been substituted for ecstasy over the years can have much worse side effects, and some of them are actually deadly. So if we're talking about the actual substance MDMA, that stands for methylene dioxymethamphetamine, a bit of a mouthful, and that's probably why the term ecstasy has been coined. So I'm a chemist, and we like to think about the structure of molecules. MDMA is a molecular substance. It's made up of molecules. The structure that I have at the top of the slide here is a representation of the shape of that molecule. It's the shape, the arrangement of atoms within that molecule that's critical to its biological functions, properties. So the O's here, for example, represent oxygen atoms. The N is a nitrogen atom. And all the kinks in the chain that you see are carbon atoms. We leave them out just for simplicity. And there's some hydrogen atoms around the outside as well. Now, you don't need to know what that structure means, but just recognise that I'll show you some other structures later where the shape changes, and it's the shape, the arrangement of atoms in those molecules that give rise to all of its properties, including, importantly in this presentation, its biological properties. So MDMA is a type of amphetamine, and it's unique in that it causes release of neurotransmitters. Neurotransmitters are small molecules that have a messaging role in your brain. They're released by neurons. They help neurons, the cells in your brain, communicate with one another. So the first one of those is serotonin, and serotonin contributes to mood and importantly has pro-social effects. It helps people connect with other people. The second neurotransmitter that is released by MDMA is dopamine. And this is associated with reward. If you have a nice meal, for example, you get a hit of dopamine, and that makes you want to go back and have another nice meal in the future. And also euphoria, and it's the intense euphoria that MDMA causes that is probably the reason why it's been used recreationally around the world for a long time. And the third neurotransmitter that is released by MDMA is noradrenaline. And this drives the stimulant activity of the drug. So it increases heart rate, blood pressure, can increase attention to a certain extent as well. So these properties of MDMA are quite unique amongst amphetamines and other drugs for that matter. And so it's actually the archetypal member of a class of drugs known as the entactogens. That word entactogen basically arises from, it's a way of describing the ability of the drug to now enable people to get in touch with their inner self. But it's also recognised for inducing feelings of emotional communion with others, oneness with a community, being able to relate to others in an empathetic kind of way, and increasing emotional openness, extinguishing some of the fears that would normally trouble thinking about distressing topics, for example. So overall, MDMA, like other psychedelics, induces an alert but altered state of consciousness, which is characterised by feelings of well-being, sociability and extroversion, interpersonal trust, and decreased feelings of fear and defensiveness. And these are ideal properties for a drug that can be used as an adjunct to psychotherapy. That's one of the areas that we're interested in with our MDMA analogues. The clinical area that's been most heavily investigated for MDMA is psychotherapy that's assisted by MDMA for post-traumatic stress disorder or PTSD. And a lot of this work has been done by MAPS, the Multidisciplinary Association for Psychedelic Studies in the States. It was recognised back in the 1980s that MDMA had these properties that lend themselves towards psychotherapy, and it was used a little bit. But then the US government clamped down their war on drugs, and it was Schedule 1, so that means it was an illicit drug with apparently no uses, and that really stymied further research into MDMA for psychotherapy, for assisted psychotherapy. Over the years, organisations like MAPS have been campaigning to do some more science on MDMA. They've actually got to the point of doing a phase 3 clinical trial. And this is mainly with post-war veterans, people with refractory post-traumatic stress disorder. Typically, they've had this for over 15 years. They've had various therapies which have been unsuccessful. So they're really in a bad way. It's really heavily impacting their life. So in a placebo-controlled trial, they were found to receive about double the benefit of people who only received psychotherapy when they were given MDMA-assisted psychotherapy. So this is a reasonable number of patients. It's like a statistically valid result and quite profound, really.
This program is going through its second phase 3 study and will likely lead to the support of MDMA being FDA approved perhaps in 1 or 2 years' time.
That leads us into telling you about what we're doing here at UWA in collaboration with Ameria. So first of all, why MDMA? Well, I've told you about some of its interesting and beneficial properties. There are some other things that make it a good starting point for drug discovery as well. So it's a small, stable molecule that gets into the brain quite readily, even when taken orally. It sounds straightforward, but that's a big problem with a lot of drug discovery for central nervous system disorders, actually having a drug that gets into the brain. The amphetamine core of methamphetamine is a privileged core in medicinal chemistry. What this means is that there are lots of other drugs that have the core, that same core, and so it's a scaffold that is a great starting point. And there are many points at which we can diversify. I mentioned earlier that the biological properties of a drug are completely dependent on the shape, the arrangement of atoms in those molecules. It therefore follows that if you change the shape, you make a small addition here or take something away somewhere else, you change the biological properties of the molecule. And there's lots of opportunities to do that with MDMA. It gives us a chance to create new molecules that have never been seen before and examine their biological properties and therapeutic potential. MDMA hits a lot of targets in the brain. By targets, I mean it interacts mainly with proteins in the brain that have different functions. And by that profile of interactions with those proteins in the brain, you get different psychological and other neurological effects. So this gives us many opportunities to tune those the selectivity of the drugs. Changing the structure affects which protein targets they interact with, and this changes the properties of the drug. So, and as I've already said, MDMA is a promising adjunct to psychotherapy, increases receptiveness and positive mood, has those unique prosocial effects that brings you closer to a therapist, for example. And it has been shown, as demonstrated by that clinical trial, that it's an effective adjunct to psychotherapy for post-traumatic stress disorder. However, those treatment sessions are quite long, and this has got to do with the long biological half-life of MDMA. So they last from 8 to 12 hours. And so this raises the possibility of maybe can we come up with some improvements on MDMA. For example, if we can adjust the rate at which it's metabolized, we could potentially have a shorter treatment. And that would mean instead of an 8 to 12 hour session, you might have a 4-hour session, which obviously is going to cost less and is less disruptive for the patient. Some of the metabolites of MDMA could potentially be toxic. That's a little bit controversial, but there is the potential by tweaking the structure of MDMA, creating MDMA analogues. Word analogues, by the way, means compounds that are structurally similar to MDMA. By tweaking those properties, we can change the rate of metabolism, potentially avoid metabolites that could be toxic. Some of the properties of MDMA are probably not necessary and, in fact, may be counteractive for psychotherapy. For example, it is a stimulant. It's got that classic amphetamine effect of stimulating, increasing heart rate, increasing blood pressure. And those components are probably not necessary, and as I said, probably counteractive for psychotherapy, where you want the patient to be nice and calm. And as I said, MDMA causes intense euphoria. I suppose it still remains to be seen whether that euphoria is necessary for its beneficial effects in psychotherapy. But if we can moderate it or perhaps remove it, we would be removing some of the abuse liability of the drug. At the moment, stocks of MDMA are attractive to a would-be thief, for example, because they might want to get in there and steal the substance because they know they can sell it on the street. If you've got a drug that doesn't cause so much euphoria, that's less of an issue. So apart from those kind of next-generation MDMA analogues for psychotherapy, there are potentially other new treatment opportunities. In addition to improving the safety of psychedelic-assisted therapy and shortening psychotherapy sessions, maybe because of this interesting profile of activity that MDMA has in the brain, by tweaking the structure, we can come up with treatments for other neurological conditions. Quick overview of how we go about our research at UWA. This is the workflow. MDMA is our inspiration. It's not necessarily our starting material for creating new analogues. So we then use sophisticated chemistry to design and synthesize new compounds. We call these MDMA analogues. I can't show you the structures of these compounds because we have to keep them confidential until we have patented. Once we've made the compounds, we've chemically characterized them and made sure they're pure, make sure the structures are what we think they should be. They go on to a series of biological screens. This starts with pharmacological screens, where we look at targets that MDMA interacts. Remember, targets are these proteins in the brain that MDMA interacts with. So we check that the analogues, we assess whether the analogues have a similar target profile. At the same time, we're looking at safety screens. There are some targets in the body and in the brain that MDMA does interact weakly with that are potential safety concerns. So when we make analogues, we're hoping to see that those interactions are lost, thereby improving the safety of our analogues. We need to look at behavioural studies in animal models to get an idea of what kind of biological activity we're getting in a complex setting, in vivo that is. And this gives us ideas about what kind of indications the compounds might be useful for. We look at metabolic assays to determine whether the drugs have a reasonable kind of half-life. If they're metabolized very quickly, they'll never be any good as a drug. If they're metabolized too slowly, that could be an issue as well. So we want that sweet spot. And in addition to neurological effects, we have recently identified some peripheral effects. These are effects outside of the brain that have therapeutic potential as well. So these studies allow us to prioritize disease indications that might be applicable to our MDMA analogues. As I mentioned, next generation versions of MDMA that are safe, that perhaps have a shorter half-life as well. Other neuropsychiatric treatments and treatments for non-neurological conditions like fibrosis. I'd just like to take you through now a case study. This is something that was done before the Ameria UWA collaboration started, but it's the thing that got me interested in MDMA as a starting point for drug discovery. And I just need to switch now. I believe some of the videos I'm going to show you are not, they're going to come out a bit jerky through a Zoom, but hopefully you get the gist of what I'm trying to show you. I got interested in MDMA because of its relevance to Parkinson's disease. I can tell you that my father suffers from Parkinson's disease, and this is what got me interested in the first place. Parkinson's disease is a complex neurodegenerative disorder. It affects the entire body, but in particular, some of the symptoms of Parkinson's disease are associated with death of neurons in a part of the midbrain called the substantia nigra. Literally, that means black stuff. These are midbrain sections taken from two deceased people, obviously. There is the black stuff, the substantia nigra, and you can see in the Parkinson's patient, it's a lot of that pigmentation is gone. So this part of the brain uses and produces the neurotransmitter dopamine. The treatment for Parkinson's disease, therefore, is to try and replace that lost dopamine, and it's done in the form of L-DOPA. L-DOPA gets absorbed, gets taken up into the brain, and then there's an enzyme that converts it into the active neurotransmitter in the brain. So without L-DOPA and without dopamine being produced normally in the brain, Parkinson's patients suffer from essentially a lack of movement. Because that dopamine helps coordinate movement, smooth movement, Parkinson's patients can't move very well. And hopefully you can get a bit of an idea of this. It's called bradykinesia, that's a technical term, a real stiffness and difficulty in moving. L-DOPA restores movement for Parkinson's patients, and it's really a wonder drug. It has amazing effects for the first 5 to 10 years, but unfortunately for most patients, during that time and as treatment progresses, they start getting some very severe side effects, and these are motor complications. That is, they're affecting movement. So this is called L-DOPA-induced dyskinesia. It's involuntary movement, and it ranges from the kind of head jerking, embarrassing kind of movements. Can potentially be dangerous because patients have falls and that kind of thing. And in its most extreme form, it can be completely debilitating. So, for example, this patient on the right, it's questionable whether there's any point in him taking his L-DOPA anymore. Without it, he can't move. With it, he's jumping all over the place and can't sit still. So that's called L-DOPA-induced dyskinesia, or LID. The connection between Parkinson's disease and MDMA arises because of this guy here. His name's Tim Lawrence, and this footage is taken from a documentary that was produced by the BBC Horizon series back in about 2000. So Tim had very severe levodopa-induced dyskinesia. We'll see examples of that in a second. And he found that when he took an ecstasy tablet, the dyskinesia was completely alleviated. So what I'm going to show you now is a small clinical trial, N=1, where Tim was given L-DOPA plus placebo, just a sugar pill on day 1, L-DOPA plus ecstasy on day 2. Now, you can see on day 1 here, he can't move at all. He's experiencing the bradykinesia, that lack of movement that's characteristic of raw Parkinson's disease. And that's because his L-DOPA hasn't kicked in yet. On day 2, for whatever reason, the L-DOPA kicks in a bit faster and he's moving quite normally. Once the L-DOPA has kicked in on day 1, you can see that Tim's experiencing that dyskinesia. He can't sit still. Whereas on day 2, I would say he's moving completely normally. Now, I saw this documentary and I found this astonishing. I'll show you some other footage as well. I thought there was something really quite profound here. And importantly, that study was backed up. This is a study of N=1, but it's been backed up with higher numbers in a primate model of Parkinson's disease. The other thing that was shown in this study is that MDMA was able to extend the therapeutic duration of L-DOPA in Tim Lawrence. In Parkinson's disease, the period of time where patients have good movement after they've taken L-DOPA is known as on time. They're moving, they're on. If we look at day one, after 135 minutes, the L-DOPA is completely worn off. He's trying to put on his jacket here, but he gives up because he can't move. On day two, at the same time point, as you can see, hopefully, Tim is moving completely normally. So that kind of profound difference between with MDMA and without MDMA is what got me interested in MDMA as a starting point for Parkinson's disease therapy. The problem with MDMA, and this was acknowledged by Tim Lawrence himself, is that it makes you high. And while that sounds okay, day to day, it's not viable. Tim said he didn't take MDMA every day because he didn't want to be high every day. So that made me ask the question, is it possible to dissociate those beneficial effects of MDMA in Parkinson's disease from its psychoactivity? So here again on the left, I have the structure of MDMA, and I've highlighted there in the magenta color the CH3 group, so that's a carbon with 3 hydrogens. We call it a methyl group. We played around altering that substituent. So you can see in the box in the middle, we've got a range of different substituents with different numbers of carbons. We then examine those compounds to see if they're psychoactive or not. The ones inside the boxed region are non-psychoactive as best we can tell based on animal models. That was done with Matthew Martin-Iverson here at UWA. He's a psychopharmacologist. Of those, UWA 101, the one with the 3-membered ring, looked to be the best. And that was evaluated in rat and primate models. And actually, it was efficacious at reducing levodopa-induced dyskinesia in the primate model. We then looked further. All of these compounds have a mirror image that is different from it, a bit like your hands are mirror images of one another, and they're different. And it turned out that the UWA 121 was the mirror image that is the most active in this case. So we call it an enantiomer in chemistry, and that single enantiomer looked to be the most promising. So in the gold standard Parkinson's disease primate model, UWA 121 extends on time, that therapeutic duration of L-DOPA, by 40% and increases the proportion of that on time that is good quality, that is not affected by disabling dyskinesia, by 215%. So it looked very promising. I'll preempt some questions from the audience about this now. This is the point where we kind of stopped this research because we didn't have a commercial partner to progress any further with, but it's something that we are certainly discussing with Emyria at the moment. And I'd just like to finish up by introducing you to the team we've got working on the project here at UWA. Dr. Jolie Lowe is mainly focusing on the analytical chemistry and data curation for the project. Dr. Glenn Kulella doing design and synthesis. And Adam Fielding's an honours student who's doing a bit of both of those activities, and he's going to come back and do some work over summer as well. The project is open to honours and PhD students, so it's quite likely that the team will grow over the next few years. That's about it from me, Michael, and I will hand back to you now.
Thank you, Matt. What a terrific overview, and I think the audience can understand kind of our excitement working with an expert like Matt and his team, who's been thinking deeply about this space, the potential for MDMA and its analogues for more than 15 years. And we're really lucky to be starting this project really from a sprinting start. So a little bit about the progress and next steps here and where we're going with this program. So far, what have we accomplished with our partnership? We've now generated more than 140 unique MDMA analogues, which have been created and characterized and have demonstrated limited off-target effects. They're those basic safety screens that Matt was alluding to before. We continue to add to that library based on the results and findings and insights from the early screening work as well. And we're also able to verify the purity and stability of these compounds to make sure that they can be suitable drug candidates. With that work is also supporting the filing of intellectual property. And recently, we've been able to launch a preclinical program taking some of the more promising compounds into studies that can help us elucidate or reveal the therapeutic potential of these drugs as well. To elicit quite profound neurological effects through the UWA-121 story. It is possible also to engineer different kinds of selectivity for different brain targets with using MDMA as the base compound. And so in the UWA-121 example, we saw increased action at the dopamine receptor or DAT, but we can also adjust the various specificity of the different neuroreceptors as well and get different kinds of behaviors, or at least that's our goal. And we know that those kinds of differences are important when we look at other drug classes that are already approved, registered, and commercialized. And so if we look at, say, the 5-HT2B, this is a serotonin target. We want to avoid agonism of this target because that can lead to fibrosis. If we can get an antagonism of that receptor, we may be able to get an anti-fibrotic activity. And that's one of the things that was revealed to us through our initial screening program. And we know that this is a really important and attractive target for a number of biotech companies. And we also know that fibrosis as a condition is a really challenging unmet need, where a couple of the leading drugs in that class generate large revenues, given the importance of that condition. But we're also seeing the ability to use this molecule to explore other therapeutic spaces as well. If we can look at increasing dopamine neurotransmission by getting more action at the dopamine receptor compared to what we might see at noradrenaline or serotonin, we may get a characterization that's similar to modafinil. This is a drug that's used for narcolepsy or to improve wakefulness in people with sleeping disorders. And we know that condition itself can also be a large and important market as well. And so on with some of these other interesting characteristics as well. Serotonin preferences, noradrenaline, and some combination drugs as well. So now we're starting to see that we have this potential maybe to differentiate this library into multiple different fields, including narcolepsy, depression, ADHD, as well as the enhanced kind of psychedelic assisted therapy potential that we discussed earlier. And our early results are already starting to show the potential to group compounds from our analog library into these subsets. We already have several leads in each of these domains, which we are now taking through that preclinical program to hopefully further reveal their potential to be therapies in these spaces, which makes this library particularly exciting, but also a source of not just one, but multiple potential therapy applications. So what does that screening workflow look like? I appreciate these are a complex slide here, but you can imagine walking from left to right, we have our synthesis and screening engine. That's the work that Matt described, 140 compounds to date, and that work is ongoing. The early screening cascade allows us to start to differentiate and group our compounds into various areas of promise, where we're looking at drugs that look and behave a little bit like MDMA, which may be more potent versions of MDMA. They become interesting. We put those in one group. Then we look at other agents which have specificity, that particular action on one or more of the key neuroreceptors, which allow us to maybe get that differentiated clinical effects, and they become another group that we're really interested in. And then we can look at some of these other targets, somewhat unexpected, but like the 5-HT2B antagonism, which may have therapeutic application in conditions like fibrosis, traditionally a non-neurological disease, but also one of great interest, given sort of the clarity of the result that we saw looking at those compounds in the screening cascade. And then we can advance those compounds into a number of phenotypic screenings. This means looking at how these compounds work in various gold standard animal models or cell lines, where we can get an idea of the actual physiological or biological effects that these drugs may have. And so a number of programs are running in parallel right now, where we're looking at some behavioral screenings and evaluations with an Australian CRO. We've got some human cell line assays, at the Institute of Respiratory Health, looking at the fibrotic or anti-fibrotic effect of some of the lead compounds from this grouping. And then more recently, you would have seen the partnership with Psychedelic, which uses really advanced computer vision and artificial intelligence to model the behaviors of different compounds in animals and compare those to reference libraries so that we can get a read on how similar our treatments look compared to other known approved drugs, and help us rapidly differentiate and identify the most promising compounds in our library. And the other study here is the metabolic screening, which helps us answer those questions around how quickly do these drugs break down in the body? Can we make predictions on what how fast they might act in the body and how long they might have their effects for? So a number of things working, looking at behavioral predictions, drug comparisons, human cell responses, and onset of action insights, which allow us to create a data package and then prioritize the most promising hits into key leads for further progression through toxicology studies and ultimately to phase 1 clinical trials. So just a quick snapshot of the Psychedelic partnership. This is a really, really interesting platform and probably worth just drawing your attention to. Psychedelic is a sophisticated and neuroscience-focused CRO based in New Jersey, the USA. They work with many large pharma companies, have had a number of successful commercial partnerships with a range of neuroscience and psychiatric-focused drug development companies. One of the great challenges of trying to develop a treatment for something complicated like a psychiatric disorder is that it is unclear exactly what kinds of action you want in the brain to get the beneficial therapeutic effects. And really the only way one can get a read on what your drug might be doing is to test that drug in an animal model and watch carefully the behavior of that treatment's influence on that animal. And Psychedelics have this really advanced, sophisticated program for that, where they can take the experimental treatment, run these animal models, and carefully measure a whole heap of parameters and data inputs, which allows them to draw comparisons between how our drug is working compared to known reference drugs, and therefore help us place where our treatments live in a therapy space, which can rapidly help us progress this program. And we're really excited to be working with such a great group like Psychedelics on this program. And so finally, what does the next 12 months look like? Well, those preclinical studies will continue to progress. We'll have data readouts, which will allow us to make further bets on the most promising treatments. We wish to select the very best leads from our library to take through to more defined proof of concept efficacy studies in animal models, which is an important first step towards phase 1 clinical trials. The expansion of the library is ongoing, and that also allows us to capture strong intellectual property around these compounds as well. So a really, really active, very generative and busy program for the next 12 months, and one we encourage you to follow along with us. And so finally, why are we so excited about this library? Well, not only do we have great support from experts like Dr. Matt Piggott and his team, we've got this, what we believe is probably one of the world's largest synthesized libraries. So these aren't theoretical, these are actually created compounds that we have access to, and it continues to grow. We are already starting to demonstrate that we have this diverse receptor binding profile, and we're able to start to characterize that, which is really exciting. And now we're starting to see some of the potential indications for these treatments, spanning really complicated and challenging conditions in neuropsychiatry, but also some other unexpected areas like fibrosis, also a major unmet need. We really look forward to your questions, and we hope that you will follow our story. Thank you very much.
Thank you, gentlemen, for the great presentation. And we encourage webinar attendees to use the Q&A function at the bottom of the screen, but I'll jump into some that have come through already. And Matthew, this one's probably for you. So where is UWA 121 at the moment?
Unfortunately, 121 has sat in limbo for some time now. When I discovered, well, when my team discovered that compound, I was a young academic, and it was important for me to publish. So I published some results on a precursor to it, and that kind of prevented it from being patented, which was a bit of an issue. But the main issue was we couldn't find the right partner to try and progress to the next step. it's called the Valley of Death, that point in drug discovery. You have an interesting lead compound that might have efficacy in an animal model, but it costs a lot of money to get it through the safety, toxicology, and similar kind of studies to the point where you can get it into a clinical trial. And at the time we made that discovery, there just wasn't the appetite for taking on an MDMA analog. It was a bit too risky. Things have changed a lot though, and the collaboration with Ameria has been wonderful. It's great to finally have an industry partner who sees the potential in these types of compounds, and they have amazing connections around the world, the resources to really make things happen. So we're not really actively pursuing Parkinson's disease as an indication at the moment, but it is something that we're thinking about, and we may pick up this story again into the future.
So just further to that then, so how far are you from phase 1 trials?
I can add to that, Matt, and you might like to say as well. I think we're at the very beginning of that work. We've got what needs to happen is these preclinical tests need to take place first. That will allow us to make more specific proof of concept animal model evaluations on the most promising compounds. And then usually if those animal models start to show promise, we would then advance into toxicology screening first before getting ready for phase 1 clinical studies. We're also using the results from these preclinical screens to improve our understanding or our explanation for why the certain structures might be having the effects they have. And so often what will happen, and Matt can talk to this too, is that we'll get an idea of what structural improvements we might continue to make in additional new compounds. We might get new ideas of where to go next in the synthesis program. So our very best leads may yet to be created, but they'll be inspired by the results from the early screens. And so likely there's a few iterations and cycles like that before we get the most promising compound possible, which we then would want to advance towards phase 1 trials. So we're likely still a year or so away or more from getting into humans, but of course we're trying to make that work happen as quickly as possible.
So what properties would you most and least like to alter from the base molecule in a commercial analog?
We're talking about MDMA for psychotherapy. Its long half-life is an issue. It increases cost of therapy sessions and is disruptive for patients. So if we had an analog of MDMA that was exactly the same except was cleared by metabolism at twice the rate, that would be beneficial. There are other things that can be improved. I mentioned reducing the stimulant activity of in an MDMA analog, probably not helpful in psychotherapy.
The question was interesting because I also asked about what qualities we'd like to preserve, I think, with MDMA, and I know you touched on that also, Matt, but one of the advantages of working with something like MDMA is you already get a small molecule that can penetrate the brain where it can actually have its effects. These compounds are typically very stable as well, which means they can be formulated into druggable agents more easily as well. So if you already get a small molecule that can get to the brain, that's already stable, you've got some of the properties that sometimes need to be reverse engineered into lead compounds, and we get those by default. And I think now we look to try and adjust the specificity of which of these brain targets we interact with.
This will sound quite academic, but we have a fascinating opportunity here as well to work out whether euphoria that's caused by MDMA and potentially other psychedelic drugs is actually a critical component of the psychotherapeutic advantage of these compounds. Is it necessary or not? I know that there are other people around the world, other companies investigating whether they can come up with analogues of LSD that don't cause hallucinations, that still cause the brain plasticity, for example. Can we come up with a non-euphoric MDMA analog that still induces these pro-social feelings that are so beneficial in psychotherapy? And they could be potentially useful in other settings. For example, a lot of dementia patients get aggressive. Could we give them a drug that makes them just chill out without making them dopey, for example? They just feel more calm, less aggressive.
So what is the likely cost of the work to get to phase 1 for each analog?
Well, although that depends on kind of the programs that we need, often there's usually not uncommon for a compound, to be somewhere between $1 to $3 million. A lot of that would depend on, I guess, how many iterations we want to do of our preclinical work, with some additional screenings required. But what we hope to do is identify a number of lead compounds with really promising preclinical data. Quite often, that's a point where we can use that data to raise additional funds or find other commercial partners to take that forward. We think by using sophisticated partnerships, advanced technology like the groups with Psychedelic, we can rapidly accelerate some of those evaluations and pick those lead compounds sooner rather than later.
Have there been any unexpected severe adverse reactions using the MDMA so far?
None of the analogs have been put into people at this point.
But with MDMA psychotherapy, I believe there are adverse reactions in some cases, and part of that possibly stems from the stimulant activity. So stimulants, if you've ever had too much caffeine before, you don't feel very good, you feel agitated. And that stimulant activity of MDMA could be a component of that. And it's really important that the people listening in recognize that MDMA has been shown to have therapeutic benefit in a controlled setting, with trained psychotherapists to oversee that kind of therapy. It's not the kind of thing that you can self-medicate on. Taking an MDMA tablet without the psychotherapy is unlikely to have benefit. And I've already mentioned, if you're getting that drug through someone on the street, there's a good chance it won't even be ecstasy.
Just building on that, currently the trials for MDMA-assisted psychotherapy and patients with PTSD require people to be off their antidepressants. It's a very high number of patients with PTSD are prescribed antidepressants, and so it needs to be really careful weaning off of that. And part of that concern is an interaction between MDMA and regular SSRIs or serotonin reuptake inhibitors. And so there is an open question: does that limit the potential of MDMA-assisted therapy for a large number of patients who are just at too great a risk to take off their antidepressants, or for whom that would be really difficult? That's an interesting sort of opportunity as well.
So in terms of commercial partners, is there FDA approval required before these talks are commercialized?
No, I don't think so. I think what that question is alluding to is the potential for the FDA approval of MDMA itself, which is imminent, and it's certainly believed to take place in the next year or two once that second phase 3 of MDMA-assisted therapy concludes. For our analogs, no, because we're sort of exploring, I guess, different neuroscience applications. They will all have their own independent kind of approval process. What I think the approval of MDMA-assisted therapy will do for our program is just generate tremendous interest in these agents, and particularly the compounds that we've identified that may have advantages over standard MDMA for assisted therapy applications.
What we have had to get approval for is not from the FDA, but from the DEA, the Drug Enforcement Agency in the US, to be able to send compounds to Psychedelic, because they're US based. We got an assessment from the DEA whether the MDMA analogs we're sending would fall under the same schedule as MDMA, that is Schedule 1. And fortunately, they came back and said, no, these are not similar enough to MDMA to fall under the same schedule. So they're all good to go.
Wonderful. And we think we've just got time for one more. So, Michael, to you, what should shareholders look forward to over the next sort of 3 to 6 months?
Particularly related to this program, I think each of those preclinical programs will have various readouts that we can, eager to share with shareholders. On the first, the simplest thing would be just the expansion of the library, the ongoing kind of growth of that library, and further results of the early screening that we do, results from the preclinical programs which we're running in parallel. We'll add additional ones and more indication-specific programs as our more promising compounds become identified in that program as well. And I think we're not too early to start approaches to various commercial partners and other groups with new kinds of screening technologies to see whether we can further advance the screening of these compounds as well. Yeah, a very active year, and that's just speaking about our MDMA program, not touching on sort of the cannabinoid program, already very advanced, working its way through phase 3, where some additional formulation work is being done as well.
Thank you so much for your time today, and thank you all for joining us for the Emyria Limited Investor Webinar. Should we have missed any questions, please feel free to reach out via the contact details on the bottom of our ASX releases. Thank you all for joining us.